A very good morning to everyone. So, my topic for today is the comparative genomics of cyanobacteria cylindrospermopsis with differential toxicities and health effects. So I thought before I get on to my specific area of research, I'll give you a little gist about cyanobacteria. So I'm sure we've all seen sites such as this in our lives at some point, whether in a lake, in a pond, so you cannot see cyanobacteria individual cells under the naked eye, but when they clump together, they form this greeny, messy thing. And so okay, we've got a cyanobacterial bloom. What's the problem? Firstly, it gives the water a dirty smell. Secondly, it ruins the lovely blue water into this dirty green thing. Also, it causes blockages in the water treatment systems, often a problem for water quality. Then, the biggest problem is the risk to humans and animals. So, cyanobacteria, it can be a skin irritant, the water. It also produces toxins, neurotoxins, hepatotoxins, cytotoxins. I'll just refer two incidences. In Palm Island, 1979, over 100 people fell ill because of cylindrospermopsin poisoning. In Brazil, 88 people died and over 2,000 were injured because of saxitoxin poisoning. So let's try and look at what causes these cyanobacterial blooms. The biggest factor is nutrients. Eutrophication basically means that the water is rich in phosphate and nitrogen and nitrates. And so let's say soil erosion or land clearings, the runoff or the water from these, when that mixes into our water bodies, it's full of nutrients, and because of that, leads to cyanobacterial blooms. Also sewage effluents, rich in nutrients. Other factors, light. Variable intensities of light help cyanobacterial blooms. Temperature, it likes warm months. Warm months, your water bodies can get blooms. Turbidity. If you look at the picture, the blue water it's got low turbidity. So turbidity means that there's stuff floating on the water because of which the sun rays can't really reach. So if you have nice, pristine, lovely blue water, that can help cyanobacterial growth. So now it's important to know that not all cyanobacteria produce toxins. You have different species of cyanobacteria. Not all species produce toxins. Even in species, not all strains produce toxins. So you can have, say, cylindrospermopsis resiboskii, the one that I work on. It has strains present all over the world, but not all produce toxins. And now we'll come on to what I work on. So this is cylindrospermopsis resiboskii. It's present in two different morphotypes. So it can be coiled in shape or it can be straight in shape. The little cells, if you look at figure 2, the little cells at the end are called terminal heterocysts. They basically help in nitrogen fixation. So if you give C. resiboskii nitrogen, it's happy. You don't give it, it can fix its own. Also, it's capable of photosynthesis. So in all, it's a very self-sufficient organism. So why do we study this C. resiboskii? Why is it important to understand how it works and its functioning? Because it produces these two deadly toxins. Cylindrospermopsin, hepatotoxin, hits your liver straight away. Saxitoxin, it's a neurotoxin, hits your brain straight away. You want to be able to know more about this organism in order to know more about toxin production and toxins. So, another reason. Now, if you look at the blue spots, between 1950 to 1990, it was a tropical organism. It liked the nice warm weather. And in the temperate regions, it was rarely found. And then, between 1990 to 2010, suddenly its occurrence in the temperate regions just significantly increased. Now, luckily so far, only in these 3 areas of the world do strains of C. resiboskii produce toxins. But you can see it's obviously adapting from a warmer temperature to a colder temperature. So who's to know, in the next 20 years, maybe it could adapt its toxin production as well. Which is why we need to understand more about this. Which brings me to my project. So for the purpose of this presentation, I'm going to call my strains A, B, and C, just to keep it simple. So I took 3 strains from Australia, Queensland, so same geographic region, and I looked at their genomes. A genome is the sum total of all the genes in the body of an organism. So I had 3 strains. One of them was toxic straight, so it produced the toxin, cylindrospermopsin, and it was a straight morphotype. The second was toxic, and it was coiled. The third did not produce any toxin, and it was a straight morphotype. So what do you think? Do you think the 2 toxin producing ones are similar, or do you think the 2 straight morphotypes are more similar to each other? And in general, do you think these 3 would be similar to each other? So we found that in size, in gene arrangement, and all the basic metabolism, like potassium metabolism, photosynthesis, they contained the exact same number of genes and the exact same genes. So they're really similar. And overall, over 90% of their genes are common, are the same. Then, between the 2 straight morphotypes, 94.8% of the genes were the same. Between the 2 toxin producing ones, 93.2% were the same. So we can see that the 2 straight morphotypes are more similar to each other than the 2 toxin producing ones. The other numbers I want you to see in this slide are 3.2, 5.5, and 2.9. Now, these are strain-specific genes, which means in A, toxic straight, the red one, 3.2% genes are present only in that strain and not in the other 2. In the blue, the non-toxic straight, 2.9% of the genes are present only in that strain and not present in the other 2. So what are these strain-specific genes and what is their function? So we found that the main function of these genes are to help the organism adapt better. I'll give you an instance. Say transport-related. Strain A, the red one, has more transport-related genes than the other 2. Maybe because where it's found, it needs to have a better transport system evolved for it to get food. Now, coming to the most important part of my project, the CYN gene cluster. Now, we already know that the toxin-producing genes contain this cluster. I'm not going to go into detail on the cluster, but so we know that C. resiboskii, this cluster is present, and it's in fact inserted in another cluster. I want you to look at the lower diagram. So you can see there's this other cluster called the HYP gene cluster, and the CYR clusters just jumped in from somewhere. Now, in other cyanobacterial species, you also have the CYR gene cluster, but it's not present in the same location. Okay, now I want you to look at the upper figure. You can see the HYP gene cluster in another cyanobacteria, Cyanothecae. Between HYPC and HYPF, there's nothing in between them. In C. resiboskii, mysteriously, this CYR gene cluster jumps and comes in. Now, this was already elucidated by, in fact, a senior from our lab before. So this is not new. So what's new about my project? Now, we knew what was happening in the toxic strains, but nobody had looked at the non-toxic strain. So we didn't know in a non-toxic strain, is the gene cluster partially missing? Is it completely missing? Is it present with mutations? We didn't know that for sure. And now we do. So if you see the gene cluster, the lower figure, in the non-toxic strain, you can see that the entire gene cluster from CYRD to CYRN is missing. only CYR0 gene is there. Now, either it could mean that it first inserted in the toxic ones and got deleted from the non-toxic, or in the non-toxic, only CYR0 jumped in. That's something we won't know. Phylogenetic markers. So you can distinguish humans on the way we look, but for bacteria, you cannot distinguish them on the way they look. And so we use phylogenetic markers to distinguish them. In general, for cyanobacteria, you use like the RPOC1, RNA polymerase subunit, or phycocyanin spacers to distinguish them. For C. resiboschii, you cannot choose them because they're identical, 100%. So there is one gene called the ITS that can be used, and we have also identified two other genes that can be used as intraspecific markers. So in conclusion, we found that the genomes of all these three C. resiboschii strains, whether they are straight in morphotype or coiled, or they're toxin producing or non-toxin producing, they're all very similar. But we found that the straight morphotypes were more similar than the toxin producing ones. We've also found that traditional phylogenetic markers cannot be used. And importantly, we found that all these organisms basically evolve or acquire genes, strain-specific genes, that help it adapt and survive better in its environment. So with that, I would like to thank the Australian Research Council, Seawater Australia, Brett and Leanne, and all my friends. And thank you for being a lovely audience. Thank you.